So today is April Fool's Day, April 1st, Wednesday, and we are going to conclude our discussion today of respiration, and then in the second half of the lecture, start on Phloem Transport and Harvest Index. So it's a two-component thing, but I don't think this will take the entire period. So let's pick up now where we left off. We had carbon use efficiency of plants, and in the last lecture I said we can separate this. This separation is particularly helpful when we model respiration and try and improve respiration efficiency in all organisms. And we separate it into the maintenance, respiration, and growth. Let's take maintenance first. Remember that, I'm going to put it way over here because this is where we had it before. Let's see. Here is our rodent, we'll say mouse. And remember, we calculated about that mouse had to eat about 50% of its weight in food every day to maintain weight. This is a mouse that's not gaining any weight, so this is respiration maintenance, the component of maintenance. Then a person was about 10% of our weight a day, and we didn't do plants. How about, here we go, get our plant on here. We have roots to maintain, we have top, green leaves in the top. A plant, what do you think the maintenance respiration of a plant is? If a mouse is 50, where do plants have to go? Nowhere. They just sit there. They don't have to move around. So the maintenance respiration of a plant can be much smaller than animals. And it turns out 1 to 3%. So plants have very low maintenance respiration. This number tends to go up when the plant's growing faster. Things chlorophyll, hard molecule to synthesize. A chlorophyll molecule only lasts about 2 days in a plant, so it has to be resynthesized. Rubisco, the enzyme wears out, it's rebuilt. That's a hard molecule to make. This is all part of maintenance respiration. So a fast-growing plant tends to inch up more toward 3%. And an old tree, let's take the Jardine juniper up here in our mountains. What's that, 1,500 years old or something? What do you think the maintenance respiration is of Jardine juniper? Probably less than 1%. That thing is just sitting there, staying alive. Really low maintenance respiration. How about houseplants? Really low light. Really, they're not trying to grow fast, they're just trying to maintain. How about, we should have a separate category right here, plastic plants. That fake plants that look like a plant, zero. They're not alive. So if something is alive, but this is a striking difference that the maintenance respiration just gets less and less. If carbon use efficiency is only about 60% efficient in our model, where's all that respiration going? It is going to make new things in the plant. So synthesizing new compounds is expensive. So we can calculate and model what it takes to synthesize new compounds. And let me put some values up here. Yeah, we better shut the door. We got a party in the hallway. This is probably, if we zoom in, you can probably see this. This is a very famous paper. It's almost a 1973 old paper The Dutch model everything in great detail, and so Penning de Vries was a famous Dutch scientist that modeled the energy it takes to make new compounds based on how complex the compounds are. And this is a long paper that I've studied many times, and I'm going to synthesize it for you. Products, requirements, and efficiency of biosynthesis, a quantitative approach. So we looked at how many bonds there were, how hard it is to make these compounds. And, I don't remember how, this is a long paper, let me see. It's almost 50 pages long. I remember studying this when I was in graduate school. But let's put a couple numbers up here, just to give you a sense of this. This now is respiration growth. So it's a separate thing here. What this is grams of sucrose, sorry, other way around, grams of product per gram of sucrose. Now, de Vries actually did this for glucose because animals use a lot of glucose, but sucrose and glucose are really close. So we take, if we were going to make sucrose in the leaves and store sucrose in sugarcane, just nothing happens. It's just made and stored. That's what sugarcane does. Guess what? The ratio is 1. Really efficient, 100%, because nothing happened. What's the next more complex compound that plants would store? How about rice seeds and wheat seeds and corn? They don't store sucrose, something a little bit more stable. Starch, a bunch of simple sugars all hooked together. I wonder if I've got my starch. It's common in cooking. You put starch in water and it's not soluble. It falls to the bottom of the jar because it's a more complex molecule. Sugar is really soluble. Starch is not too hard to make. We can make a gram of starch. We can make 0.9 grams of starch with 1 gram of sucrose because it's simple. Now we get more complicated. What are cell walls made out of? Cellulose. Harder to make than starch. 0.8. Now we've got the non-structural, well, we've got the structural carbohydrates in here. We've made cell walls, we've starch. Now we need to do the next amino acids. Harder to make. I don't have these numbers all memorized. I gotta look at my notes. 0.6. Amino acids are a lot harder to make than cellulose. Now all the little amino acids join together to make proteins, which are big molecules, hundreds and hundreds of amino acids. So we got proteins. 0.5. 2 grams of sucrose to make 1 gram of proteins. We keep going. Now we're into 2 things that are really hard to make. What is mostly in the trunk of trees that makes wood really rigid, a 2x4? Lignin, yeah. I should show a picture of lignin, but it is a huge molecule. It's so hard, a tree falls over in the forest and takes the microbe 50 years to degrade the lignin in the tree. It is so complex and so rugged. We can't digest lignin, we can't digest cellulose either. But lignin's really tough. It's a carbohydrate, but it's a huge complex molecule. And lignin is harder to make than proteins. We have one final compound. What kind of oils do we use for cooking in a kitchen? All plant-based oils. Safflower, corn oil, olive oil, sunflower oil, all oils. Lipids or oils, very hard to make. They're very reduced. They're high energy compounds, but it's hard to make them, and lipids are about 0.4. So now you see where we end up with 60% efficiency for carbon use efficiency, a little bit to maintenance, much less than animals, but a lot more to growth. And plants are a combination of all of these things. They're proteins and lipids, the lipid bilayer. Plants got to synthesize that lipid. Some plants, like say lettuce, not much lignin in lettuce. So lettuce can be more efficient because they're not making hard compounds. And a tree is less efficient because they've got to make a lot of hard compounds. If we take this, and this is another thing that Penny DeBreeze did, I'll put it, this is precious space, but I'm going to put this up here. We take the same concept and do seeds now, grams of product, we're going to put seed type. And we're going to put RG, growth efficiency, over here. The staple seeds for the whole world, rice, wheat, corn. Let me put the categories up here because you're going to see where we're going. These are one category. Most of the weight of rice and wheat and corn? Starch. They can be very efficiently synthesized. They have some protein, but not a lot. When we take the synthesis efficiency, 0.75. So that averages all these things. They have some lipids in them too, but very little. Now we go to soybeans. And other crops like soybeans. What are soybeans famous for? What do we use them for? Soybean oil. They're high lipids. They're also high protein, much higher than rice and corn and wheat. So correspondingly, soybeans are lots harder to make. And this number falls to about 0.5 for soybeans. Now let's take something really hard to make. I had to look this up. Peanuts. They're really high in lipids. I think peanuts are 45% lipids. Soybeans are about 20% lipids. So peanuts, this number drops all the way down to 0.35. So there's the theoretical concept. It's more than theoretical. The analysis of how difficult it is to make all these compounds for a plant, and then the integrated number, what we can get from seeds from those plants. And it gives you a sense of maintenance and growth respiration. So when we say, oh my peanuts have low yields, oh my soybeans have low yields, compared to, say, especially corn, Well, yeah, they're making a lot harder compounds. Corn, all I had to do was make starch. It's got it easy, so the yield isn't much bigger for corn. What we should calculate is the energy yield from crops, because these compounds, remember, let's put this right here. If we do, one more time, carbos, proteins, and lipids. This is on the side of every single food product you buy in the grocery store, and you can calculate the kilocalories per gram from each of these. And believe me, if this was a human nutrition class, the first day you'd get and memorize these numbers and have them. Remember what these are? It's pretty easy. And kilocalories per gram, 4. Now, proteins should be a higher value because they're harder to make, right? Takes more energy. But proteins are less digestible by people. It takes energy to digest proteins. So for energy for people, proteins come in at 4. Now these are rounded to 1 digit because there's different kinds of carbohydrates. But 4, now we get to lipids, boom, 9. Lipids have 2.25 times as much energy per unit weight as carbohydrates and proteins for human nutrition. That's why you try to eat less lipids. We all eat way too much lipids anyway, but they sure give you a lot of energy. I guess some bar, and if you're starving, shouldn't be a protein bar. It should be a lipid bar, high energy per unit weight if you're backpacking. But lipids are harder to digest too. You can't digest them as fast. So here's how hard it is for plants to make, and then the value for human nutrition. Now, when we model this, we can take this back to relative growth rates. Maintenance respiration is both of these are like a tax. This maintenance respiration property tax, you pay it on what you have. If you own a big house, you pay a lot of real estate taxes. Growth respiration is like an income tax. You can live in a pup tent, and if you make a 6-figure salary, you still got to pay a lot of income taxes. So this is a real nice analogy, and most people say pay taxes on both of these. But property tax is plant size, plant mass, or plant, and income tax is plant growth. I'm going to say plant growth rate. Now, when we model this, how do we get plant mass and plant growth rate? RGR, relative growth rate, grams of new product per gram of old per day. And if we have coefficients for growth and coefficients for property tax, we can put those two numbers into a relative growth rate and calculate the amounts of respiration from that. So let's do that. We can take a young plant growing fast, which is going to have a lot of income tax or a lot of growth respiration, and an old plant growing slowly, which is going to have a lot of maintenance respiration, but not much growth respiration. And to do this, by the way, is part of this week's homework. So to do this, This here is just a written version of what I've just put on the board. It helps to look up typical values for these. I already put plant maintenance respiration. Let's see, let's put it right here. This is times respiration maintenance, and this is, it can be really tiny. I put, where did I put it? 1%. Yeah, let's just put 1 to 3%. And actually for really old trees, it's even less than 1, so it's tiny. This, sorry, the old is times maintenance respiration, 1 to 3%. The new is times growth respiration. And you can see the average. If you're making a lot of lipids, it's low. If you're making starch, so we often put 30% in here for synthesis of new. So if we know the relative growth rate, we know the ratio of these two numbers, and we could calculate the weight of carbohydrates used in respiration for each per each day. And that helps us back calculate what can we do to make plants more efficient. Can we help them have less maintenance respiration? Can we help them have less growth? Maintenance respiration, you're probably wondering, how do we measure that? The most standard way is take a plant in lower light and bring it down until it's not growing. It's really like putting a plant at the light compensation point and say, how much light does it take to have zero growth, to just get up to zero? Let's see, let's put that right up here. Remember our graph, PPFD, the light input, here's photosynthesis, here's respiration, right there. That point, light compensation point. The amount of light it takes to get to zero. And photosynthesis right here is all going to maintenance respiration. So you can see a big fast-growing plant that might take a lot more light to get to zero. And a house plant hardly takes any light to get to zero. But this is maintenance respiration. That point right there. Then the rest of it is growth respiration. So the homework asks you to take some relative growth rates and take some typical income tax rates, the fraction of carbon going to growth, fraction going to maintenance, and calculate what's going on with the plant. And to help you with that, let's work through an example problem. I suppose I could have put this grid on the board, but I think we can see it right here. This just reviews everything I've said, and I'll send you this as a handout. But here's the equation. Growth respiration calculated on growth rate, and maintenance on plant mass. So now let's take this. There's a table, but let's take that table and blow it up. Great. Now we can see it. Now I need to find a pencil, which, if we're lucky, there's one in here. All right. I'm not going to work every one of these, because if we do one, you'll get it. You'll see them all. We're going to take a 10-gram plant. So this is a young plant growing really fast. Growth rate 0.25. This is what everybody's calculating on COVID now, this exact number of growth rate. But remember, 0.3 is a really fast-growing plant. So this is a young, healthy, fast-growing plant. The next category would be a medium-aged plant, growing much slower. And the final one would be like a tree, something that's really, really growing slow. But let's just work the first one. So let me put over here this is P net, and this is respiration dark. Now this doesn't give us any indication of growth and maintenance. It just gives us the total respiration. But P net for this plant is really delta W. It's how much weight did that plant grow in a day, and it's the weight of the plant, 10 grams W, times the relative growth rate, 0.25. So if it's 0.25 times 10, this number is 2.5. So that is the absolute amount of growth this plant grew. Now it's got to pay maintenance respiration. So in this example, you can see it right there, 1.5% is what we're assuming. So somewhere in between these numbers, it had to pay maintenance respiration on this 10 grams of growth. Not the growth rate yet, just the 10 grams of growth. So this is 0.015 times 10, so this number is 0.15, because it's 10 grams of plant, and that's all these units are in grams. The amount of photosynthesis so a 10 gram plant, 0.15 grams went to maintenance. Now let's do growth. Now we're assuming 30%. So pretty typical value for growth respiration. So this growth respiration tax is paid on this new growth right here. And this comes out to be 0.75 because it's a tax on this. So 30% of that number is 0.75. This right here says P net integrated. So this is the P net over the whole day. That's what integrated means. And it's shown right here. Integrated P net was the new gross. So look, we have 0.15 grams going to maintenance. We have 0.75 grams going to growth. The total respiration is the sum of these. RM for maintenance, RG for growth. This comes out to be that plus that is 0.9 grams of carbohydrate that went to respiration. Now, remember how we did growths in here, where we had this going to here, and we did gross photosynthesis. Gross is how much it grew plus the respiration. So this is 2.5 plus 0.9, 3.4. Total, this is total gross photosynthesis. The plant had to fix 3.4 grams of sucrose to gain weight of 2.5 grams at the end of the day. This young, fast-growing plant. Now, can we do carbon use efficiency of this plant? Sure we can. Carbon use efficiency is the weight gain divided by the total. So it's net gain divided by total. So this is 2.5 over 3.4, which is 73% carbon use efficiency. Really high, but again, this is a young, fast-growing plant. And now these final two columns is maintenance respiration percent, and that's maintenance respiration divided by this total here, and that number comes out to be 4% went to maintenance for this plant, which is maintenance right here divided by total, 3.4. And the other one, growth respiration, comes out to be 22% for this plant. This is an example of how we can model and separate growth and maintenance respiration and calculate how plants are doing in a more detailed way, depending on what they're making. Let me tell you how these come out. I worked these out. I'm not going to work them, but this one is going to be less. 63%, 100-gram plant growing at 0.05 growth rate, so it has more maintenance. This is now the maintenance in this plant jumps up to 19%, and the growth is also 19%. And finally, we get to a much bigger plant growing really slow, and the carbon use efficiency drops to 36, but 54% is maintenance and 11% is growth. Boy, you can barely see that on the edge of the screen. So this is maintenance and growth respiration and how we break it down to better understand what the potential is for respiration. In conclusion, for this whole respiration section, having worked on this a lot in this lab and with colleagues around the world, it is hard to improve respiration efficiency. Cold nights help a little, but 10, 20 years ago, we thought we had lines with low respiration, and that turned out not to work. We're still trying to breed plants that are more efficient for respiration and create environmental conditions that are better for respiration, but it's not easy. And in general, if we say 60% respiration carbon use efficiency in our model, I think the best case scenario, we're going to get that maybe 10% better, maybe 0.66 instead of 0.6, but that's still a huge improvement to crop productivity. So now let's look at the next part of this. I guess I should draw this in a different color. Now we're going to move to Phloem Transport, carbon partitioning, and harvest index. This is the fourth and final component of our model. Photon capture, quantum yield, respiration efficiency, and now harvest index. And harvest index is heavily dependent on Phloem Transport. We have bred plants for enormous fruits, and the ratio of fruits to stems and leaves has now gone way up, and it's starting to become limited by how fast we can move stuff in the phloem. So in the book, this is the next chapter, transport and partitioning. In plant physiology textbooks, there's a chapter on Phloem transport. Every plant physiology textbook ever written on the principles of Phloem transport, how fast it moves, how it moves. It's wonderful to study because we really understand the physics of Phloem transport. Plant physiology textbooks never discuss harvest index though. And once we move all this carbohydrate around in the plant, how much can we store in the whether it's the roots or the seeds or wherever we're interested in getting it. So this is a hybrid between the basics of Phloem transport and the more applied aspects of harvest index. So Phloem transport is pressure. It's the same thing as a tube of water and flow of water in a tube. Summer is coming. We're going to have the garden hoses out. You got pressure pushing water through a hose, and you have flow rate, and it comes down to a flux equals a driving gradient times a conductance. The equation that everybody's already good at using this. So we can measure driving gradients, and sometimes we measure flux, and then we get a conductance for the phloem, because it's a solution flowing through a tube in the plant. But like a lot of things that we do, most things, the first step is calculating what's the driving gradient for pushing this along. And if we can double the driving gradient, we're going to double the flux. The conductance is really related to just the diameter and the number of the tubes, of the conducting elements. And we can measure those under a microscope, or we can measure these two things and calculate conductance like we've been doing before. But let's take a look at driving gradients now. This is pressure. What's pressure in physics? Pressure is force per unit area. Pressure, let's see, let me give myself some room. This is important, so we're going to draw it big. Equals force area. That seems so simple, but it's more complex than it seems. If you had high school physics, you would have learned F equals ma, maybe college physics, mass times acceleration. So this is a little more complex, but this is the force that we push on things. And that's a nice analogy. And now it's times area. So the force per area. Let's take an example. If you've got a decent quality fridge and you open it and you look at stuff, then you close the door, you can't get the door open again right away. It's sucked shut. What's going on? The warm air got cold, contracted, made a negative suction for as cold air is more dense, and it sucked the door shut, and you go, wow, that's because the area of your refrigerator door is enormous. So it took a lot of pressure on the handle to pull the door back open. That's a concept of pressure. If there's a large area, you can have a lot of pressure with the small force. How do we measure pressure? What's the SI unit for pressure? This is a case where SI units really help, even though we don't use them very much, but we should. The SI unit is PASCAL, the Pascal, named after Pascal, who did all the work. So do we use that in anything? How about Soil Science 101, and around here it's Soil Science 3000. We learn how well water is bound to soil. That's where this whole thing starts. The plant has to get water out of the soil. So let's put this right over here in yet another color. Soil. Field capacity. First of all, how do we measure the suction on soil? There is a case where we make nice use of SI units, and there's a certain suction associated with field capacity. It's easy to get water out of soil when it's fully wet. It's wet, but it's drained. And the high end, I'm not going to put high, I'm going to put wet. And when it's really dry, what do we call a really dry soil? What's the limit at which plants can get water out of soil? There's a name for that. PWP, permanent wilting point. I won't write the whole thing because you can. Permanent wilting point. This is the worst word because it's not permanent and it's not the wilting point either. It should be a temporary water stress point, but it's roughly the level at which plants can't get water out. Now this matters. We're talking about phloem transport here, but this directly matters in xylem transport because this whole thing starts with water getting sucked up into the top of the plant and it's sucking it out of this dry soil. So here is the $64,000 question. How do we measure? Pressure and suction are the same thing. One's positive numbers, one's negative numbers. So we're either sucking water along in the xylem, or in the case of the phloem, we're pushing it along. But these are still the driving gradients for fluid flow, for solution and water flow in plants. So what's the unit we use? If you took this class 20 years ago, we would have said bars. And that's not an SI unit. And it took soil scientists several decades to get rid of bars. And I think it's mostly gone from textbooks. Even though it's a fine unit, it's just not SI. What do we use for SI units? MPa, MPa, megapascals. Now, this is covered in every soil science book and people write it down in their notes, but rarely can people remember these two numbers. These are negative numbers because it's a suction. How much suction does it take, how much negative suction, negative pressure does it take to get water out of wet soil?
0.03.
Oops, 0.03 out of soil. How about permanent wilting point?
Minus 1.5.
Yep, minus 1.5. Now, we're going to build on these numbers for a whole plant. How hard does a plant have to suck? How much can it push? But now these numbers become quantitatively important. You can calculate all kinds of things from them, and it starts with the amount of water in the soil. This is a little, not good analogies to people because we just drink water, and a glass of water is 0. If the field capacity was not drained, this number would be 0 up here. These are both negative numbers because they're suctions. All right. Now, I've asked this question many times to physicians and to lots of people, and I'm always making analogies to human metabolism and human fluid flow. We haven't the humans and plants. Here's the question. It's very tricky and it's a bit counterintuitive, but it helps you understand pressures. Which system has the higher pressures? Pressures or suctions, people or plants? You cut yourself, blood squirts out. You cut a plant, nothing squirts out. So that means people must have higher pressures to move fluids around in our body. We have a heart, boom, 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 pumping. Plants do not have a heart. They have to use osmotic gradients to suck fluids around, which we are coming to. But let's get back to this question, which has the higher pressures? And I've had fun with this over the years because I ask it to a lot of physicians and they go, whoa, let me see. But then they can't do the plant part of it anyway. But they always think people have the higher pressures. When you go, oh, for a prop, I should bring in a blood pressure cuff. Every time you go to a physician, the first thing they check is your blood pressure. You donate blood, they check your blood pressure. Anything, blood pressure. You can buy blood pressure kits for home use. In fact, if I would have thought of it, I would have brought mine at home and I could have measured my own blood pressure. How much is blood pressure in people? What's the numbers and what's the units? Even though everybody gets it checked all the time, they're harder to remember than this. But what are the units? Let me see. I'll put it right here. Here's a person. Blood pressure. We got to give them a face. Now, over the years, we've had some nurses in the class, and they're pretty good. They're better than doctors at remembering numbers of things, I think. There's 2 numbers on this. And you will really remember these when you get to be 80 years old because your blood pressure goes up and you worry about it and people take medications to keep it down. But there's 2 numbers here. Remember them? 120 over 80. This is called systolic and diastolic pressure. This is the pressure right after your heart beats. You get a pulsive pressure, and then you get a resting pressure. Boom, 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 boom. It's going back and forth. This is ideal. It's kind of like the typical body pressure with no fever. 98.6 Fahrenheit and 37 point something Celsius standard. This is the standard for a healthy person. Here's the $64,000 question. And you can look this up on the internet pretty fast. What are the units with this? Is this megapascals? Oh my, no. The medical profession is not into SI units at all. It's based on mercury, because that's the old school way to measure pressure, was a column of mercury. And 100 years ago, before we had fancy gauges, this is millimeters Hg, which is mercury, both of these, millimeters mercury. Is this a bigger number than this? This isn't plants yet, but this is the suction they have to work with, pretty big range. You can answer this question by looking up conversion factors for pressure on the internet. And think of all the units of pressure now. And once you start to get your head around this, you can become bilingual in like 12 languages if you get the units of pressure. When you look up pressure if you watch TV, the weather forecaster, he's talking about a high pressure front's moving in or a low pressure front's moving in. What are the units they use? It's pressure. It's a ratio of force and area. So often they'll use inches of mercury. I don't have space to write down these. Inches of mercury, inches of Hg, and then there's millimeters of mercury. Now this translates into force per unit area because it's the force to draw mercury up in a column. I don't have a picture of this, but the early barometers had a little dish like this, and then an upside down column, and they would put mercury in here, and it, depending on the suction, it would get drawn up in this column, and that distance right there is delta P. And the pressure gradient, and there was tick marks on here. That's where inches of mercury came from. For low pressures, we use water. We use inches of water and centimeters of water. How about your bicycle tires? We pressurize those. PSI. Force, pounds, area, inches squared. So PSI is yet another example of a unit of pressure. They're all over the place. Let's see, did I bring? I had. Here it is. Let me put this one up here. This is a conversion book. Atmospheres, that's another, how many atmospheres of pressure that we have. We go into outer space, there's no pressure, so 0 atmospheres. Sea level, it's 1. At our elevation, as you know very well, it's 0.85 atmospheres. And that's pounds per square foot. Feet of water, inches of mercury, atmospheres, kilograms per centimeter, oh my goodness, short tons per square foot. This whole thing's a mess. But if you just learn the SI units and you get everything to SI, then boom. So it's the first step's unit conversion. Here's another thing I looked up on the internet. Here's another little table. You get these to convert from these units, multiply by this to get these other units. Now we get some more. Torr. 1 torr is 1 millimeter of mercury. Here's pascals. A pascal is, I don't have much room left. What's the unit of force in the SI system? Not pounds, it's newtons. And what's the area? Meters squared. So a newton per meter squared, we can look it up on here, is, well, you can convert to everything, but one Pascal. Newton per meter squared, 1 Pascal. On and on and on. There's a lot of units. But we're going to work strictly in SI units, but it's still useful to think about this concept. Now, a couple minutes left. Let's take, I will send you, you can look this up on the internet. It's hard. I wonder if I need to send handouts, but that's all right. If you got the handout right in front of you, we're all looking at the same handout. Where's my pencil? Now let's answer this question. Plants or people have the higher pressure system? We don't know yet, but we know the units. And the plants have even more suction than the soil. So plants are at least 1 megapascal of suction in them. And is that bigger or smaller than millimeters of mercury? This is a huge conversion table. Clear this off. And it's way too small to read. And I could give this as a handout. This is coming. This whole side is low pressure, and it's all it is a lookup chart with units here, and you go back and forth, and this column is high pressure. So let me skip to this, and if we go down here, to, it's going to be too hard to see, 121 inches of mercury is, you got to believe me that I read this chart right, 21 kilopascals. And obviously 80 is less than that. 80 is, 80'd be up here. This is 10.7. 10, roughly 11 kilopascals. So someday we'll get all the blood pressure in the world converted to SI units, 21 and 11. That's the pressure in our systems. Now, plants, wow, megapascals. So plants have much higher pressure and suction systems than people. And it's a good thing, because if we were a high pressure system, you cut yourself, you'd bleed to death. But plants have a lot of suction, and then against that, they create a gradient to push things through the phloem. And as you'll see in the next lecture, we push through the phloem and we suck through the xylem. But both of those move all the fluids around in a plant, just like they do in people. But this is just an interesting question for plants and people and getting the units together. I've got another minute here, a few minutes. Let's let's go way over here, because I got clean space. We'll keep this up. When you calculated, here's our plant. Another plant right here. Roots, leaves. Here's a leaf. And here's a stomate in that leaf, like that. And we calculated water coming out of the stomates, the water shown in blue. And remember, we had delta H, and we calculated the driving gradient for water vapor movement out of a stomate. And the units, without knowing the details, the units for that were in kilopascals. But down here in the soil, we're drawing water out of roughly -1 megapascal. This is 1,000 times bigger suction than this gradient up here. How can this be? It's all SI units, so we can make nice comparisons. But if the water is this tightly bound, why are the stomates, the final guardian of water in the plant, moving it out to the air when the gradients are only in the order of kilopascals? I have to erase I got to erase my mouse. There. This is water vapor. This is a gas, water vapor gas. This is liquid water. Water vapor follows the ideal gas law, PV-nRT, and it's by diffusion. Diffusion of gases in air, and we calculate the driving gradient pressure, partial pressures. This follows, liquid water follows different principles than gas. Now, in the next lecture, I'll show you how the equation to convert. But liquid water, if you ever had any previous class with water potential, this is chemical potential. Chemical potential. And the universal symbol for this, drawn large, is the pitchfork symbol, psi. So anytime we talk about liquid water, we're talking about chemical potential principles, and we measure it in water potential, psi. This is the gradient of gases as it comes out of a leaf. So obviously the conversion's a big deal, because a kilopascal of water vapor, the leaf might be at -3 megapascals of chemical water potential in the leaf, and the water is still moving out the stomates really fast as a gas in kPa. So that's the difference. Water potential and gas movement, partial pressures of gases. And as soon as you see the conversion equation, you'll get that. This one is the gradients over here are delta psi. This one, the gradients, we just call them delta H for humidity. So this, in conclusion, this is called, this very famous thing, footer in red. the soil, plant, atmosphere, soil to plant, plant to atmosphere continuum. And part of the mission statement for the Plant, Soils, and Climate Department is to understand water movement in the soil, plant, atmosphere continuum. And the whole name of the department is Soils and Plants and Climate. And our department links these, and we model this on all kinds of scales. Cellular scale, we're going to do it in whole plants. The climate scientists are looking at this in a big scale in the atmosphere. Soil scientists just look at the soil and the water movement in the soil. We're going to link them all through the plant to the atmosphere. And that's the subject of the next 2 lectures. I'll stop here and we'll pick this up Friday. But one more thing, I have a homework. We're a little bit behind on homework, so I combined a couple principles into one homework. This is coming in your email. Due Monday. Now, it's Wednesday today, so you have some extra time, but when you email them to me, I can grade them and get them back to you by a week from today. The first one is calculating yield potential. I think you'll have fun with this. Here's the world record holder of giant pumpkins. He said he grew this in a 30 by 30 foot area in his backyard. Is it even possible he did that without cheating with steroids and hormones? And you calculate what the components of yield would have to be for him to grow that pumpkin in his backyard. How efficient would it have to be? And there's room to show your work. And then part 2 of that is, if he had CO2 enrichment, and if like he built a tent in his backyard, now how big could the pumpkin get? What could he have grown? So show your work on these, just like the take-home exam, send it back to me. This is a 2-pager, because we got 2 things. Part 2, respiration and carbon use efficiency. Here's the data. Calculate the carbon use efficiency of these plants. They're in a bag, so we're tracking photosynthesis and respiration. And then from today's lecture, here's 2 plants, old plant growing slow, young plant growing fast. Calculate maintenance, growth, and carbon use efficiency of those plants. Hopefully these aren't too difficult. You just got to look at the handouts and apply the principles to a different kind of question. But that's how you learn to do this, is by practice of calculations. So you have 5 days to send these to me by Monday, and I'll grade them and send them back to you. We'll talk to you again on Friday.